All right, we're going to revisit chapters 16 and 17, which is addressing the respiratory system, and we're leaving off really tying into the cardiovascular system. These two systems are very much interrelated with one another. So even though we're in the respiratory system, we have to address the circulatory system because the circulatory system transports our respiratory gases. And we left off with the basic concept. If I want to move a gas in the body, I have to create a what? A gradient, and more specifically a pressure gradient. You could say chemical concentration gradient, but we're looking at gases, and so we do measure them in pressures, and that's how we can identify the directionality. And if you look at it in a simplistic view, it makes sense. If I want oxygen in my tissues, the pressure for oxygen should be what in those tissues? Lesser than where? In the blood, and then less than in the, and even less in the lungs, which is less than the atmosphere. So we have to have a gradient so that gas, and more specifically oxygen, would be able to diffuse down this pressure gradient from where it's in greater pressure in the atmosphere, where it's in lesser pressure in your tissues, so it will move down there. And we're looking at specifically a single gas. We talk about that gas as partial pressure. We're just really kind of honing in when we look at the partial pressure of a single gas. We noted as well last week that the pressure gradient is reversed for carbon dioxide. So that carbon dioxide is the greatest pressure inside of your tissues than it is in your blood, than it is in your lungs, than it is in the atmosphere. Okay, so carbon dioxide would fuse down its pressure gradient out of your tissues, out into the atmosphere. So that pressure would, that gradient would actually be reversed. What we're going to do here is now just apply the values of that. So bigger number, bigger pressure, lower number, lower pressure. And obviously the gas would move right from its greater to its lesser pressure. Now realize, if a gas moves to an area, what happens to that area of that pressure for that gas? If I move more of a gas into something, the pressure in that something would have gone up. Okay? But if we look in the blood is just transporting the respiratory gases. And so when it's being, after the exchange happens between the systemic capillaries or the pulmonary capillaries, the pressure shouldn't change. It should be pretty consistent. So once we add oxygen in the systemic circuit, it'll stay that way until it's exposed to a gradient, and vice versa for carbon dioxide. So if we look at the pressure gradient for oxygen, as the blood is entering the pulmonary capillaries, the pressure in the capillaries, the pulmonary capillaries, should be really low. Because the blood's already fed into the systemic circuit, it's already fed the tissues, if you will, and so the content has declined significantly. So blood returning to the lungs has a partial pressure of about 40 millimeters of mercury. In the blood. The air entering into the lungs, it's coming fresh from the atmosphere, so the pressure of oxygen should be really high, at about 100 or 104 millimeters of mercury, so a high partial pressure. We can clearly see a gradient of about how many millimeters of mercury? About 60 to 64. Okay, so a huge, steep gradient, about 64 millimeters of mercury, basically pushes oxygen from the alveoli into the pulmonary capillaries. That will clearly add oxygen to the blood, and our blood would then return to the left side of the heart for distribution, right out to the systemic circuit. It's a huge pressure gradient, and what it does is just pushes the oxygen into the blood. But if you remember, on Thursday, we talked about the solubility of the gases. We learned that oxygen is not very soluble, so we obviously have a transporter. What's a transporter? Hemoglobin. But to get it to the transporter, we have to push it. And the way we're going to push it is this huge steep gradient of about 60 millimeters of mercury. So we push it in there. And it allows for the blood to go through the capillaries. Even though we said the velocity is declining, it's still fast enough so the blood's not having to wait in the lungs, clotting up, whatever, waiting for oxygen to get in. So just push oxygen into the pulmonary circulatory system. Does that make a little bit of sense? The blood's flowing, and we're just pushing pushing oxygen in. We learned that the gradient is reversed at the pulmonary capillaries for carbon dioxide, but we'll notice that the gradient is not near the magnitude of that of what we see for oxygen. What's the gradient for carbon dioxide? How many millimeters of mercury? 6. 6 millimeters of mercury. In the pulmonary capillaries, as blood is entering, the pressure of carbon dioxide is about 46, and in the lungs it's about 40, so we have a gradient of about 6. It's significantly less than that of oxygen. And that goes back to what I was just elaborating on a moment ago, is carbon dioxide is more or less soluble than oxygen. More soluble, so we don't need to push it forcefully. It's going to go into the blood plasma or out of the blood plasma a lot easier compared to oxygen. And so these pressure gradients allow for the gases to be exchanged at the appropriate rates to maintain homeostasis for each of those gases. Can y'all see the reverse gradient at the lungs for each gas? The blood would obviously transport the proportion of gases, and those gradients would be reversed again at the tissues, at the systemic tissues. The pressure for oxygen in the systemic circuit would have a, in the capillaries leading up to the systemic circuit would have a pressure of about what? About 100 millimeters of mercury. But in the tissues, about how much? About 40 to give us a gradient of about 60. So significantly lower in our tissues, which makes sense because if our cells are metabolizing, they're consuming the oxygen. Right? So they're going to actively create that gradient there for oxygen. And then the reverse is true for carbon dioxide. The pressure for carbon dioxide should obviously be higher in your tissues and less in the systemic capillaries if we're making oxygen, if we're using carbon dioxide. So it's just reversed. What questions do you have about these gradients that push, these partial pressure gradients that push the gases and where they need to go? Everybody comfortable with it? Concept? I want oxygen in my tissues, so the pressure should be in my tissues. If I want oxygen in my tissues, the pressure should be lower, lesser than in my blood, than in my lungs, than in the atmosphere. If I want carbon dioxide out, pressure is greater in the tissues than it is in the systemic circuit, than it is in the capillaries, than it is in the alveoli, than it is in the atmosphere. What questions do you guys have over that movement? This is about where we left off on Thursday. We were talking about the transport of our respiratory gases, and I had you guys look ahead on that image of the end of your handout. And we learned already that oxygen is not very soluble, which was kind of implied when we said there's a special protein transporter. Ties back to chapter 4 and 5 material that we studied earlier this semester. Something not very soluble, give it a carrier using a protein. Well, oxygen's not very soluble. It has a protein carrier called hemoglobin. And that's a preferred route for the transport of oxygen to bind with hemoglobin. However, oxygen is not completely repulsive to water. So it can be partially soluble in oxygen, in your liquid watery plasma environment, but not very well. So very modestly, around 10%, somewhere around there, of your oxygen can be transported in the blood, dissolved, not bound to anything. But the high proportion, 90-some-odd% of your oxygen is going to diffuse into a red blood cell to be bound to hemoglobin. Because we know that each red blood cell has about 150 million hemoglobin molecules, so we can make high efficiency for oxygen carrying capacity. If we look at some technicalities of hemoglobin, if hemoglobin is bound with oxygen, it's called oxyhemoglobin. You can abbreviate HbO2. And the hemoglobin may be reduced if it lets go If it releases oxygen, it's got to bind something, and it will tend to bind. What do you think? What can it bind? Hydrogen ions. So in that case, we would call it a reduced. You can see the nomenclature there, HE, the reduced form. And we also noted as well, at the end of chapter 15, we learned that hemoglobin can reversibly bind and unbind. So it just depends on the status of the tissues, whether or not hemoglobin is going to bind or let go of oxygen. That's all that little shorthand note is illustrating the reversible binding and unbinding of hemoglobin. How many binding sites does each hemoglobin have for oxygen? 4 binding sites. If all 4 binding sites within a hemoglobin molecule are bound with oxygen, that hemoglobin molecule is considered to be saturated with oxygen, or it may be partially saturated if one or more of the hemoglobin groups are not bound with oxygen. So either fully or partially saturated with hemoglobin. With oxygen, excuse me. We can further study this. If you remember, we said that oxygen can reversibly bind with hemoglobin, and the follow-up question is, well, then what is it about our body or hemoglobin that allows for it to let go or bind oxygen? And where do we need oxygen? Where? In our tissues, our cells, right? Our muscles, our brain, right? Everywhere. In our tissues, oxygen is used, and most specifically, right, during the electron transport chain. So, metabolically active tissues consume oxygen. More metabolically active, more oxygen consumption, and vice versa. Right? We kind of studied that with the little rat experiment like a month ago. So, thinking along those lines, what characteristics did we say about tissues or organisms that have increased metabolism? What do we see in the rat? Okay, so there are more heat released, so temperature would go up in tissues that need oxygen. What else? Tissues that are active are gases. They're doing what? They're making CO2, so our partial pressure for CO2 is probably going up. If our partial pressure of carbon dioxide is going up, when you think about that reaction that we learned in lab, what's going to happen? Our pH changes so it becomes a little bit what? Acidic. So, pH drops a little bit, carbon dioxide goes up. What about our oxygen levels in a tissue that's active? In the tissue, the oxygen levels are actually what? Using it, so the oxygen levels in the tissue would be less. All of those factors and some additional factors actually contribute to hemoglobin changing shape and letting go of oxygen. So, if my partial pressure of oxygen goes which way? Up or down? Down can cause hemoglobin to let go. My temperature, if it goes up, change the shape of that protein, let go. My partial, let's go over here. My pressure of carbon dioxide probably goes up, thusly causing the pH to go down. All of these factors contribute to hemoglobin from letting go of oxygen. And if you look at the effect is, it lets go of oxygen, oxygen goes to the tissues, right, to keep the tissue metabolizing and alive and healthy. Questions? Okay, I'll stop here. Alright, so, I mean, if a tissue is metabolically active, it needs what? Oxygen. So all this byproducts of consuming oxygen, favor hemoglobin, and letting go, so more oxygen is delivered, the tissue can continue to metabolize. Those things are summarized here. We said, if the cells be metabolically active, glucose, right, for example, carbon dioxide being produced, which drops our pH, and our temperature is up. All of those factors contribute to hemoglobin from letting go of oxygen so it can meet the demands of those tissues. Or it's described as unloading. The oxygen is unloaded into the tissues. So oxygen can be carried one of two ways. One way, the preferred route, is what? Bound to hemoglobin. And we know now hemoglobin can let go when it's in areas where it's needed. Or oxygen may be carried how? Dissolved in the blood plasma as oxygen. There's two ways in which oxygen can be transported. Carbon dioxide can be transported how many ways? 3 ways. It can be how? What'd you say? It can be dissolved in the plasma, converted to bicarbonate ion, and did I hear it? Bound to hemoglobin. So we've got an additional mechanism for transporting carbon dioxide. So a small proportion of it as well can be dissolved in the blood plasma. And we also noted last week that hemoglobin can bind not just oxygen, but carbon dioxide and other molecules. So we understand that. And when it's bound to hemoglobin, the proper term is called carbaminohemoglobin. But we focused on the third mechanism as well in lab last week. We learned that the preferred route, or the greatest proportion of our carbon dioxide, actually gets converted into a more soluble ionic form, which is called bicarbonate ions. So about nearly 3/4 of your carbon dioxide will undergo this transformation to form bicarbonate ions. We understand from all semester that ions, right, they have the charges, they're water soluble, so it's a lot more water soluble in this form. As well as, when this happens, what else are we fixing, if you will? What else is being regulated when we convert carbon dioxide into bicarbonate ions? pH of the blood. So it's kind of like a twofold benefit, if you will. Not only is it more soluble, but it contributes to maintaining our blood pH. Just reviewing the formula, we already covered this in lab, so I'll just kind of briefly walk through it. We've learned that when carbon dioxide is present, it can interact with water to form an intermediate molecule called carbonic acid. We also learned that this reaction is this molecule is kind of unstable, and so as soon as it's formed, it'll dissociate into its ionic form, into hydrogen ions and to bicarbonate ions. What we clarified in lab though is this can happen by 2 mechanisms. So this third way of carbon dioxide conversion, or this carry, can occur in one of 2 ways. What were the 2 ways in which this reaction can occur? What do we call it? How could this happen? This reaction. Where, how? With an enzyme that is compartmentalized where? In the red blood cells. We learned that this reaction can actually be enhanced when carbon dioxide diffuses into a red blood cell. Anybody remember the name of the enzyme? Carbonic anhydrase. Thank you. Carbonic anhydrase can enhance the production of carbonic acid in the red blood cells to produce bicarbonate ions and hydrogen ions. So we learned this can be sped up, if you will, by utilizing the enzyme carbonic anhydrase inside of a red blood cell. Is that the only location where this reaction can occur? Yes or no? No. In what part of the blood? This was in the red blood cell. What part of the blood would the in the plasma. In the blood plasma, the reaction can still occur, but what? It's slower. Why? It's not enzymatically enhanced. So it'll just happen, described as being spontaneous. It'll just spontaneously happen, but it takes a little bit longer versus a reaction that's utilizing an enzyme. Either way, right, carbon dioxide can be converted to bicarbonate ions using this mechanism. Notice, though, the reaction is what? It's reversible. So, when these bicarbonate ions get to our lungs, we don't exhale bicarbonate ion, right? We have to convert it back to form carbon dioxide. When that reaction occurs inside of the red blood cell, that bicarbonate ion will get transported out of the red blood cell into the plasma where we said it's more soluble. We don't want to overwhelm the inside of the red blood cell with a bunch of extra product. But our bicarbonate ion is charged, the same as a blood cell, not a nerve cell, not a muscle cell, so we want to maintain neutrality, basically. We don't want it to generate an action potential or something strange like that. So we pump out a bunch of negatively charged ions. We should pump in the negatively charged ions. And the ions that are pumped in are called chloride ions. So it's just a 1-to-1 ratio. And when that happens, when bicarbonate ions pumped out, chloride ions pumped in, it's called the chloride shift. Same thing with that. It should help maintain neutrality. So the chloride ion moves in to the red blood cell to maintain the electrical charge of the red blood cell. So, is the bicarbonate ion trapped in the red blood cell after it's formed? It's actually what? It's what? Moved out. So the bicarbonate ion, even though it was formed in the red blood cell, it's going to be transported in the what? In the blood plasma, in the plasma component in the blood. These reactions are summarized for you in those images that I had you guys look at over the weekend. And so these two images summarize the last 45 minutes or hour discussion that we've had between the last 2 days. So if you look at it, give me a heads up, the first image is where in the body? Generically at a what? Tissue, some systemic tissue. We can, even if it wasn't labeled, we would know it's the systemic tissue because why? As if it wasn't labeled as such. You know, the tissue cells weren't labeled, the blood plasma. How would I know that this is at the peripheral tissues? CO2 is going out, or going out of the tissue into the blood. Oxygen is going out of the blood into the tissues. So we can see the directionality. This image summarizes the ways in which the gases are transported. We learned that oxygen is transported how many ways? 2. The arrows are proportionate, so the preferred route is being bound to hemoglobin. Over the evening, you can go back and add to what we've learned the factors here that cause hemoglobin to let go. Like what? Temperature, pH, pressure gradients for each carbon dioxide and oxygen. You can add those in. You can also add the actual pressure values. The partial pressure of oxygen in our blood is about higher than that. The gradient is about 60, so it's about 100, 104. In our tissues, it's about 40. So you can just put all that information summarized here in this image. We also learned that carbon dioxide can be carried in how many mechanisms? How many fashions? 3, but we see how many arrows here? 4. Why are there 4 arrows in those 3 ways? There's 2 different ways to do the bicarbonate ion formation. So we learned that the preferred route is for carbon dioxide to diffuse into our red blood cells interacts with carbonic acid, anhydrase to form carbonic acid. This image also does a great job illustrating how the bicarbonate ion is transported out and the chloride shift balances the cell. However, we learned that bicarbonate ion formation can occur in the red blood cell or out of the red blood cell. So those bicarbonate ion is one way that we can convert to carry carbon dioxide. We also learned we can just dissolve, and we also learned that it can bind to hemoglobin. What questions do you have at this point about kind of putting the gradients, the activity of the tissues, and mechanisms for transport kind of concisely together? That looks so excited to talk about these gas transport. The next image that we have in your handout is just the reversal at the pulmonary capillaries. And so as we identified as well, everything's reversed. If bicarbonate ion goes back into the red blood cell to enhance its conversion back to carbon dioxide, we've got to do what's called the reverse chloride shift. The chloride ion goes which way? Out, and bicarbonate ion goes into the red blood cell. And then it'll get converted back into what? Here. And water. Good. Alright, so the next image, we can understand that it's at the pulmonary capillaries because we can see the alveoli. So what type of cells are there in purple? Type 1. Type 1 cells are the cells that make up the bulk of the alveolar structure. So you could do the same thing here, reversing the pressure gradient, so going right down the partial pressure of those. You could still see that oxygen would load preferably into a red blood cell. Same thing with carbon dioxide. We do not exhale bicarbonate ion, so it has to be transformed back to carbon dioxide. What questions do you have at this point? On the mechanisms for transporting each oxygen and carbon dioxide? As we know different sensors. I don't have this image for you, but I want to go ahead and present it to you about controlling respiration. What do you think probably controls respiration? What kind of mechanism? Yes? Our autonomic nervous system, right? Respiration is controlled autonomically. You don't have to focus on breathing when you're deep in sleep, right? And so, reflex arms, right? Sensor, control center, effector organs, our ribs, and our intercostals, our diaphragm. What do you think our body might monitor to regulate respiration? CO2, O2 level, pH. Those are very important to regulate. Can you think of something else outside the box that might cause you to change your respiration rate? To what? I don't know if it was your by the medulla oblongata. As you guys noted, we got to monitor our blood conditions, our oxygen, and our hydrogen ion content, and carbon dioxide. This may be where you're going, like activity of the body, increased glucose, etc. As well as just you feel something in your lungs and your respiratory system, and you try to get that foreign particle out. But it's a neural reflex that primarily controls respiration. Which hopefully makes it autonomic nervous system to control your breathing. You can certainly override your breathing patterns like we did in lab, but you don't have to consciously regulate each tidal button. It's very normal. I don't want you guys to worry about writing anything down. If you shoot me an email, I'll send you may have an image like this in your textbook. I just don't see the image reference on it. Let me back up to make sure you have the notes for the previous section as well. Just restating what we learned about carbon dioxide at the pulmonary circuit. Everything's got to be reversed. Bicarbonate ions got to get converted back to carbon dioxide, etc. I'll look to see if there's a similar image in your textbook and give that page reference to you guys about the control of respiration. What I'd like to do is go ahead and transition to the renal system, which we're covering in lab this week. And as I mentioned, I'm kind of going to go off the cuff a little bit and show you an animation which will summarize a whole heck of a lot about the renal system. So what I would do as a student is take notes and or get your pre-lab out, just kind of verify your labeling. So we're going to go through the structures and obviously the what? Function. Our renal structures. We're going to look at, obviously, the gross anatomy of the kidneys, but we're here in physiology, so we really need to know the microscopic structure so we can understand the real function of the entire kidney itself. And so, how many kidneys do we have? A normally well-developed person would have 2 kidneys, and they both do the exact same job. Which endocrine glands are associated with your kidneys? Adrenal glands. We also learned that the kidney themselves have endocrine ability by producing renin, which regulates eventually our blood pressure, which we'll talk about on Thursday. The renal system is, or you can say the urinary system, you can call it the renal system, is comprised of more than just the kidneys. Other structures in the renal system include your ureters, which we're going to transport your urine from the kidney down for storage into the bladder. The bladder just is a temporary storage site, and then any urine would be removed to the body through the urethra, to the male or the female structure. You notice that the kidneys are not parallel with one another. One's a little offset. Okay? The left kidney is a little bit higher than the right kidney. And if we look at the structure of the kidney, it, like a lot of other structures in our body, have an outer, it'll have what's called an outer cortex and an inner medulla. So going along with that caution that I've been giving you guys all semester, if you say cortex or medulla, you really got to give some other additional anatomical hints to figure out what you're talking about. If we look at the basic structure, there is the cortex, and it's just kind of this thin layer, maybe like half an inch or less, this outer cortex of the kidneys. Let me see if I can this area that's highlighted in the kind of red pinkish color is referred to the cortex. Adjacent to it would be the medulla. The medulla is described as being made of the pyramids, and if you look closely, you kind of see some kind of triangular structures. You see those in there? And so that would be the medulla. And then the innermost portion is actually making up what's called the pelvis. Okay, so not bony matter, but it's called the renal pelvis. And it's the renal pelvis is going to collect the actual urine and then transport it to the ureters for temporary storage down in the bladder. We're going to spend our time between the cortex and the medulla. That's where the urine is actually going to be formed. Okay, so we've got to look at those structures. Because the kidneys are a reconditioning organ, we've used that word before. Kidneys are reconditioning organs, so they're going to have a very high vascularity. They're very well vascularized. So renal artery is going to supply each kidney. Renal vein would drain each kidney. You'll notice how interspersed will get within the kidney itself. And so that's kind of the gross anatomy of the kidney. We obviously want to zoom in and look at the functioning bit of the kidney, and these areas are called what? What's the name of the functioning structures of your kidney? So what? Part of it is called, that's a specific structure within this whole thing. The whole thing, she said glomerulus, that's part of it. The whole thing is called a nephron. And so what I want to look at in better detail is the nephron. So if you have your pre-lab out, we can label this. Now, does every person in the whole world and all of history draw everything exactly the same? No. Okay, so on this nephron, it's almost like a maze. So whichever image you're looking at, the one in your lab manual, the one I have presented here, the other ones that I'll show today, you've really got to scrutinize to get some anatomical locations. Then you can figure out the structure and the physiology of that structure. So we're going is, the nephrons are three-dimensional structures in your kidney. There are millions of these in your body, estimated about 1 million per kidney. Okay, so this is just really zooming in on one tiny microscopic structure. Whoever drew this could have rotated it, 360 degrees, and stopping at each degree, right? So it's just a three-dimensional structure. Now, if we look at the nephron, we said it's very well vascularized, so it has a lot of blood supply to it. Let's review our blood vessel structures. Our primary vessels, things taking blood away from the heart, are called arteries. Those lead into arterioles, which lead into capillaries, which lead into venules, which lead into veins. Artery, arteriole, capillary, venual, vein. That's normal. We're going in, and everything's got a big butt, right, in physiology. But in the kidney, the vascularity of the nephron is slightly modified. So there's not one capillary bed, but how many? Two. There's two capillary beds. It's how many sets of arterioles? Two. So in the kidney, we're going to see the vascularity go artery, arteriole, then arterioles lead into capillaries, then another arteriole, then another set of capillaries, then a venule, then a vein. So the kidney, at the nephrons, we're going to see artery, arteriole, capillary, arteriole, capillary, venule, vein. And so we've got something kind of unique here. Because what's the job of the kidneys? To filter. If we go back to any other capillary, capillaries did what? What do they do? The transfer, we said the processes are first, bulk flow is comprised of 2 steps, including there's a pressure gradient which drives it definitely. What's the process? I'll just set it a moment ago. Okay, so what's the term used to describe it properly? Collectively bulk flow. So reabsorb. At the kidney, we first want to just do what? Of those processes. We want to only filter, not reabsorb initially. If kidneys filtered and reabsorbed, we would never form what? Urine. So we have to have this weird mechanism, this unique mechanism, for some capillaries to only filter, not reabsorb. Okay. So the first set of capillaries in the nephrons are designed so that they filter only, so that we can form something called filtrate. That's what we'll modify to make urine. The filtrates will be carried in a set of structures called tubes. The blood would be carried in the blood vessels. The filtrate would be carried in the tubes. But to get it to the tubes, we've got to first make it. So the capillaries have to first filter, not reabsorb. Okay, so keep that concept in your mind, that the capillary structure in the nephron is unique. That arterial capillary is because we first want to filter only, prevent any reabsorption. And so the way that the kidneys are built allows for that. So, what's what then? If you look at these, it almost looks like a tangled mess, but everything's obviously built, right, to interact with one another. So the blood vessels are working with the tubes, so they're going to be surrounding and working with the tubes. In this image, the blood vessel supply is colored red and purple and blue, illustrating to and from. And the tubes are kind of the yellow-orange type structures. Okay, so you with me so far? Blood supply in the vessels, filtrate in the tubes. So basically, we're going to follow two fluids. Blood going one way, filtrate going a different way. But collectively, just trying to make urine. Go with me? Kidneys eventually make urine by sorting off our blood plasma. So if you look here, where do I start? Well, for me, I go for some landmark structures. What do you think is a landmark structure in a nephron? The what? The glomerulus. Okay. What'd you say? The renal artery. If it's colored, you can see that. Okay, so that would be a good starting point too. If you're looking at grayscale images, it's difficult. If you're looking under a microscope, it's kind of okay. So, but if you look at the glomerulus, it's really characteristic. It's this globular looking set of blood vessels, and more specifically capillaries. And so if you can find this glomerulus, then kind of work backwards, investigate backwards, you can kind of get some anatomy. Remember, the nephrons, artery, arterial, capillary, arterial. Almost messed up on you. Artery, arterial, capillary, arterial, capillary, then medulla and vein. Sorry about that. So here, I've got one set of capillary bed, my glomerulus, and then I can see another set of capillaries all over around the tube. The question is, what's what? Well, we can apply our terms afferent, efferent here. So we're obviously not looking at nerves, but we can get some directionality. Afferent means towards, efferent means away from. The towards and away from is the glomerulus. And it's talking about arterial that bring blood to the glomerulus, or blood that takes blood away from the glomerulus. And so to find the glomerulus, I can see some arterial. Can you see this? Can you see these two structures in my hand? Those are either the afferent or efferent arterial. The question is which? Well, the afferent arterial brings blood to the glomerulus. The efferent arterial takes blood away from the glomerulus. And then it's the efferent arterial that serves the peritoneum, or serves the tubes, right? It's the next set of capillary beds. So looking at these two, the top one is which one? There's quite an enunciation. So efferent, the top one is the efferent, that's taking blood away. I'll walk you through. Okay, so away from the glomerulus. This one on the bottom is the afferent arteriole, which is taking blood towards the glomerulus, which we can see it's coming right off of the renal artery. So it's the first arteriole that goes to the glomerulus, which would be the afferent arteriole. The reason I know it's not the efferent arteriole as well, yes, because the next capillary bed. The arteriole between the glomerulus and the peritubular capillaries is the efferent arteriole. That little vessel between the glomerular capillaries and the peritubular capillaries is the, which one? Efferent arteriole. Which, yes, would eventually be entering into the venous supply. Afferent arteriole brings blood to, efferent arteriole takes blood away from. So if you have your pre-lab or your lecture notes, why don't you locate afferent arteriole? And then just put an arrow to illustrate where the blood would be going through that vessel. Just to identify the directionality of the blood. And then maybe label that as afferent or efferent. If the lecture note is labeled for you, just add an arrow. Can you see this shunt that's highlighted? Which arteriole is this? Afferent. So if I was trying to write on here for you, the blood would be traveling to the glomerulus. Blood would then enter into the glomerulus and then be drained by the efferent arteriole. And then blood would go into the what? What are they called? What are the capillaries called after the glomerulus? They're called peritubular capillaries because they happen to be around right next to the tubes. The efferent arteriole is not individually highlighted. This is an efferent arteriole that would drain into the peritubular capillaries. So let me just repeat. The blood supply of a nephron, these tiny little microscopic structures, first gets to the nephron via an afferent arteriole, which brings blood into this specialized set of capillaries called the what? The glomerular capillaries. What happens at the glomerular capillaries? Filtration only. Filtration only happens at the glomerular capillaries. There is no, better not be any reabsorption. The glomerular capillaries are going to form our, what do we call it? We're going to call it filtrate at this point. That's a good note though. At this point it's filtrate. It's not definitely urine. We're going to work it, if you will. We're going to add to, take from, etc. But we don't call it urine yet. We call it filtrate. Any blood that is not filtered remains obviously in the circulatory system. So it goes from the glomerulus to the efferent arteriole, then to the peritubular capillaries. The peritubular capillaries are like normal capillaries, where they may what? They may filter, and they definitely have a lot of reabsorption going on. kidney tissue itself alive. You know, keep the kidney alive. It kind of serves a need. So blood flow to the nephron enters via the what? Afferent what? Arterial. It goes into the glomerulus, which is a specialized set of what? Capillaries, where we have filtration only taking place. Forms of filtrate, which we'll come back to in just a minute. I want to stay in the blood vessel, though. Any blood that's not filtered enters into the efferent arterial, then into the peritubular capillaries, which we'll get to in just a minute. That's the blood supply. We obviously care about filtrate, where it's collected and where it goes, because eventually it's going to be used to make our actual urine itself. So where is the filtrate formed? At the glomerulus. So we have to have a catch net, if you will, to catch the filtrate. There's not a net, set of cells to make this cool structure. What is that structure called? The Bowman's capsule. The whole thing is called the nephron, but the part that actually captures the filtrate is called the Bowman's capsule, highlighted here. You notice these cells, they surround the glomerular capillaries, so it's kind of a covering, if you will. Or filtrate, that's blood plasma that's filtered, right, forms our filtrate. And fluid will be pushed down this entire set of tubes, so isolated fluid, our filtrate. It's going to go on its own little journey. As the filtrate goes through the tubes, what's happening? It's now to the blood vessels, goes to the tubes. What? It's definitely going to be a pressure gradient, pushing it down, gradient. What? What's going to happen to our filtrate? We're going to reabsorb some substances out of the filtrate, put it back into the what? Blood. Which part of the vessel? Reabsorb. Which one? Peritubular capillary. So our filtrate's going to go through these tubes, and our kidneys are going to start picking out, or these nephrons are going to start picking out useful molecules, like glucose. We don't want to put it out, right? So let me do. What about amino acids? We like those. Let's not peel it out. Don't have any working. Makes sense, right? Hopefully. What about ATP? Right? What about calcium? Pick it out, right? And so what happens is the filtrate travels through the tube. We selectively reabsorb useful substances, or maybe do what? Push out more waste or excessive. What's the name of that directionality? In the kidney tubule, where we're now adding substances back into the tube. You can see that. Reabsorption takes it out, there's a process that puts it back in. Called we first filter it. You only need 3 steps, or your 3 steps. You first filter it, then you can reabsorb. The next step is to secrete. So as the filtrate's flowing through our kidney tubules, we reabsorb useful substances like glucose. Glucose is the primary one, like picking out, and other amino acids, etc. But as the filtrate flows through, anything excessive in the interstitial fluid can actually be secreted back into the filtrate. It's wasteful or excessive, something like that. Like most commonly you've heard of urea. It can get secreted back into the filtrate. And at some point we're no longer manipulating it. It's the end of the journey and we call it what? Urine. But as it's flowing through the tubes, we call it filtrate. Before formation of it is the process, is filtration, but the stuff that's being transported is filtrate. So let's go through and look at the tubes, because each structure of the tube is going to have a specific job, whether it be reabsorption or secretion of particular substances. So the filtrate is first trapped where? At the glomerulus. It was formed by the what? Which capillaries? Glomerular capillaries. Form the filtrate, and the filtrate's trapped now in this big structure called the Bowman's capsule. Filtrate's just high pressure, the fluid flowing through, almost like your hose flowing, pushing out water. So filtrate is flowing through these tubular structures. After the Bowman's capsule, the filtrate enters into the first area of the tube called the proximal. Proximal means what? Proximal means closest. It's the proximal is the tube's closest to this capsule. The proximal convoluted tubule, you kind of see why it's called the convoluted tubule. See how twisted it is? We're twisting because we want to what? Increase surface area. What do you think happens at the proximal convoluted tubule? Whole heck of a lot of which process? Reabsorption. The proximal convoluted tubule is the area of the tube and the nephrons where we see a whole bunch of reabsorption happening. That's where we first pick out glucose, salt, sodium, chloride, you keep naming it, they're reabsorbed back into the peritubular pathway. We want to keep those things. After the proximal convoluted tubule, the filtrate will enter into an area called the what? The collectively, the whole thing is called the loop of something, the Henle, which has two ends. One end is called the A or D, okay, so ascending or descending limb. If we look at it this way, the first area, the filtrate would be moving which way? Descending. Let's just describe this loop of the limb. So filtrate would be flowing, in this case, down through the descending limb of the Henle. And if you're looking at your lab notes and your lecture notes, the images are inverted. Remember the three-dimensional structure. So in your lab notes, I believe it's the structure on the right-hand side. You follow through all the proximal convoluted tubule. And on the right-hand side, use the right-hand side. Good. Yes. So in your lab manual, the right-hand loop portion of the loop is the descending limb of the loop of the loop. In your lecture notes, I think it's inverted. You have your lecture notes, they're drawn backwards. Either way, you follow it through. So from the proximal convoluted tubule fluid is going down, the filtrate's going down, so we call that the descending limb of the Henle. Then it hits the loop and then it travels up, and so we call that the ascending limb of the loop of Henle. The next structure is called the what? The distal convoluted tubule. We've gotten really far away from the glomerulus, right? If you're following, we're getting far away, it's called the distal convoluted tubule. Where there's still some what going on? Reabsorption, but we now see a high proportion of secretion happening at the distal convoluted tubule. The filtrate would then exit the distal convoluted tubule and enter into the collecting duct. And if you look closely, the collecting duct collects filtrate from multiple nephrons. It's just kind of the common tube that we're dumping in there. At the collecting duct, there is modest, very minimal secretion and reabsorption. It can still happen, and not near the magnitude like you see the rest of the tubular structure. So it still can happen, but so we can still change the filtrate, so technically you can still call it filtrate in the collecting duct. The collecting duct would then drain into the what? Well, directly into the renal pelvis, and then into the ureter. So by the time it gets to the pelvis, the renal pelvis, we can go ahead and call it what? Urine. We're no longer exchanging. We're no longer secreting or modifying or anything like that. Yes. Anytime it's trapped in the tubular structures, filter. Because it would be inappropriate to call something that's first filtered out waste. Because we know that all of this stuff that our body needs is going to be reabsorbed. So it's not plasma, it's not your blood vessels anymore. It's not urine, because it's not your pelvis, but it's all filtered. It's trapped in our tubes. This will hopefully help by looking at this oversimplified animation of filtrate formation. This is taking that complex looking nephron, stretching it out. The peritubular capillaries, unfortunately, are not shown in here, but they would be associated with it. And so anytime we see something going in or out, it eventually go into or come from the capillaries. This structure over here is clearly the pulmonary. It's in there, so what's surrounding it? The Bowman's capsule, which leads into the proximal convoluted tubule and the descending limb Henry, then the ascending and the distal convoluted tubule, and now our collecting duct. We're just going to see movement, right? Filtration, reabsorption, secretion of substances. We'll see some substances that require active transport. We'll also look at the reabsorption and secretion of salt, like sodium and chloride ions, the ones that we care a lot about. Water, nutrients like glucose, waste products like urea, there are excessive products like potassium. So these are kind of what we're going to see playing along the way. The first process to eventually make our urine, first the blood has to be what? Filtered at our capillaries. So we can't forget capillary dynamics. Are red blood cells filtered out? No. Are big, large proteins filtered out? No. Okay. Is 100% of our blood filtered out? No. A high proportion of it is, but still we have some exiting via which structure? That label is off. Okay, so I have blood entering the afferent arteriole. Some is filtered, but the other is pushed out via the efferent arteriole. I want you to make a mental note. Look at the diameter between the afferent and the efferent. Okay? We won't have time to talk about it today, but on Thursday we'll address it. Filtrate is formed and first enters into the proximal convoluted tubule because it's trapped by the Bowman's capsule. We can see our first round of what? Well, those are nutrients. Reabsorption. What type of transport is this? It's active. We're using ATP. So active transport for reabsorbing. As well, passive transports, mechanisms, channels, etc. could have been dropped, but they're the same. Or reabsorbing. Anything that's not reabsorbed, retained in the tubes. Well, if we look at the proximal convoluted tubule, you can still see more reabsorption right going on. We're just kind of, if you will, picking out the good stuff, keeping it, reabsorbing. The refined filtrate will then enter into the descending limb Henle, and you can see what it's specialized for. What is the descending limb Henle specialized for? Reabsorption of water. Okay. I know it's reabsorption because the water is moving out of the tube into my capillaries. Okay? So it's being reabsorbed into the capillaries. Okay? The filtrate would then travel to the ascending limb and loop at Henle, where we still see some reabsorption, but now it's a little bit more specialized for what? Our salts, like sodium and chloride, for example. We know that those ions are important, and so we can hormonally control that here at the ascending limb and loop at Henle. We can see some active transport, reabsorption of it as well. And then the filtrate would enter into the what? Distal convoluted tubule, where we still can certainly see some reabsorption going on, but we also begin to see some what? A higher proportion of secretion, where we see waste products being transported back into the filtrate, for example. So you have being added into the filtrate, being secreted. The blue and the yellow are going from the interstitial fluid back into the filtrate, so it's being secreted into the filtrate. What's the question? The difference in secretion versus filtration is going to be those products may have originated, well, usually very precisely regulated. It would be ideal if these cells were drawn larger. This is looking at something that's traversing multiple cells. But these transporters are in the tubules themselves, facing the lumen of the tube. So it's physically passing you out. Right? So even though it may have originated in interstitial fluid or even originally been filtered, when the kidney tubule itself specifically transports something from the kidney cell into the tube, it's called secretion. Yes. It's specializing in anything lipid soluble being secreted as well. This is lipid soluble. We need some of our hormones, pregnancy tests, maybe. Where do you get it? You know, it's a standard blood test for you to just scan it. Okay, because your hormones are, or we could say we could super secreted. Or lipid soluble. They're everywhere. All right.